All right, welcome to part 4 of translation, where we actually talk about translation. I know it's been quite a wait, so by the end of this lesson, I hope you will be able to outline the structural feature of ribosomes, outline the 3 stages of translation, and describe the steps that occur during initiation, elongation, and termination during translation. All right. So, translation occurs on the surface of a large macromolecular complex that we call the ribosome. Now, bacteria only have a single type of ribosome, but eukaryotic cells have several distinct types of ribosomes. And this is all due to its organelles that have an odd evolutionary past. So, eukaryotes have one type that is found in the cytoplasm, and it is the main ribosome that actually belongs to the eukaryotic cell itself. The other types of ribosomes are what exists inside of the mitochondria and inside of chloroplasts. If you recall, both mitochondria and chloroplasts have their own genome inside, and they in fact also have their own ribosomes. This is an artifact of the fact that both of these organelles used to be intracellular symbiotic bacteria. All right, so both of these used to be bacteria, and if you actually look at mitochondrial and chloroplast ribosomes, they have a lot more similarity to bacterial ribosomes. So from this point forward, whenever we refer to eukaryotic ribosomes, we are referring to the ribosomes in the cytoplasm, the ones that actually originate from the genomes of the eukaryotic cell itself. Now, a ribosome is a complex structure. It is excessively large. It is one of those molecular complexes that are visible on electron micrograph. Most of the times you cannot see a protein complex by itself. There are a few exceptions. Ribosomes are one of them. There is a large and a small subunit to a ribosome, and both of them contain both proteins and rRNA. So, we've talked about the fact that there is transfer RNAs and that there are messenger RNAs. Well, these are ribosomal RNAs. And we talked about the origin of some of these ribosomal RNAs when we talked about RNA processing, because a lot of them are produced as a single RNA and then trimmed into smaller pieces. All right, in bacteria, we have the small subunit, which is called the 30S subunit. This is a reference to how quickly or to what level it sediments in centrifugation. So this S is a unit of sedimentation, and it refers roughly to its molecular weight, but also size as well, or shape. Now, the small subunit has 21 different proteins and has a large piece of RNA called the 16S rRNA. Eukaryotic small subunits is a 40S in size, so it's a bit bigger, and it contains 33 proteins, and it has again a single chunk of RNA. It's called the 18S RNA. Let's move over to the large subunit. The large subunit is called the 50S in bacteria, or the 60S in eukaryotic organisms. Bacteria have 34 proteins and 2 distinct chunks of RNA, so a 5S and a 23S. Eukaryotes have 49 different proteins in this large subunit and contain 3 distinct pieces of RNA. The completely assembled ribosome is oftentimes referred to when we make a distinction between bacterial and eukaryotic. So this will be the 70S subunit, or the 70S ribosome, and the 80S in eukaryotes. Now you may be wondering, well, this is 70, isn't 30 plus 50 80? Well, again, this is referring to how quickly the ribosome will fall through liquid, in a specific liquid, under centrifugation. So so molecular weight is definitely one aspect of that, but it's also the overall shape. So combined together, it doesn't quite hit 80, right? So it's called a 70S. So this is a physical property of the whole thing. It's not something that can simply be added up by its pieces. All right, in eukaryotes, it is an 80S, and you can see all the various pieces. I would, if you are interested in medical school, I would keep these numbers in your head because they tend to come up in things like the MCAT. So, during bacterial translation, you've got the messenger RNA. It is more or less laying on the surface of the 30S subunit. And as the polypeptide is being synthesized in the 50S subunit, it is exiting through a little channel here. You'll notice 3 distinct sites inside of the 50S subunit. We call them E for the exit site, P for the polypeptide site, and A for the aminoacyl site. So this is essentially where a transfer RNA comes in. It gets its amino acid gets added on to this growing chain. It then moves over and it continues to extend out. It then moves over into this exit site, and that releases the spent transfer RNA. Now, there are 3 stages involved here. So there's the initiation process, there's the actual elongation, and finally there are particular steps in the termination of translation. All right, so here we have kind of the entire cycle, and we'll go through some of the particulars of each step. You can see here that we have the large subunit, the small subunit of the ribosome. We have a messenger RNA that is sitting here, and we also have our very first initiating transfer RNA with its amino acid. During the initiation step, these things are all combined together. There are also proteins that are involved in bringing all these complexes together. Then during the elongation step, as I said, you see a new amino acid, or a new transfer RNA, charged transfer RNA with its amino acid coming into the A site. The growing polypeptide chain will then be transferred onto this amino acid. And then both of these 2 transfer RNAs will be shifted over one. So this transfer RNA, once it has been cleaved from the amino acids, it will transfer to the E site, and the amino acid that just gains the growing polypeptide chain will get transferred to P, and then we get a new site in there, and we repeat that cycle, and the spent transfer RNA that is in the E site will get emptied out. At some point, we will reach a stop codon. At that point, a transfer RNA is not available to bind in that site, so instead, a release factor will bind into that site. That will cause the growing polypeptide chain to be cleaved from the transfer RNA in the P site, and then the whole complex will fall apart, and we will have a completed polypeptide. So let's stop and go through each one of the steps: initiation, elongation, and termination, and we will kind of compare and contrast bacterial and eukaryotic systems. They are very, very similar. It's a highly conserved system, but there are a few distinct differences out there. So let's start with the initiation stage, and we'll specifically start with bacteria and then show you the differences in eukaryotes. So the messenger RNA and an initiator transfer RNA and the ribose subunits will form an initial complex, but this complex requires some specific initiation factors. Now, the initiator transfer RNA will recognize the start codon of the messenger RNA. In bacteria, this is a unique transfer RNA, which is designated transfer RNA FMET. This stands for N-formyl methionine. This is simply methionine that has formaldehyde attached to it. Formaldehyde is simply a carbon double bonded to an oxygen and a hydrogen, and this has been added on to methionine. So there are transfer RNAs out there in bacteria that they start off getting methionine added, and then some of those have been modified to this formyl methionine. And in bacteria, this formalized version of it is required for the initiation of translation. Now, the start codon is, of course, AUG, but in bacteria, you can also sometimes use GUG or UUG, particularly when they are part of what we call an operon. And I'll discuss more about that later. But there is a little bit of flexibility. But regardless of what that codon is, it is putting in N-formyl methionine. All right, so this all kind of starts out because in the 30S subunit, there is a 16S piece of ribosomal RNA. It has a particular sequence in it, which is complementary to a sequence that we call the Shine-Downgarno sequence, which is always immediately upstream of the start codon in messenger RNAs. So in bacteria, messenger RNAs, when you have a start codon, will always have this Shine-Downgarno sequence up above it. The 16S rRNA is complementary to that. So the messenger RNA is directly binding one of the rRNAs in the 30S subunit. All right, so here we have the 30S subunit, within it, there is the 16S piece of our RNA, and initiation factors 1 and 3 will bind into the E site and A site portion of the 30S subunit. This complex will then bind a messenger RNA with the Shine-Dalgarno sequence binding to a short stretch of the 16S rRNA. Next, initiation factor 2 will come in and bind initiation factor 3, and this, while it is bound to GTP, will recruit the initiator transfer RNA. So this is, again, N-formyl methionine transfer RNA. Once this complex is assembled, initiation factors 1 and 3 will be released. Initiation factor 2 will hydrolyze its GTP and be released. So this is another unit of energy being burned. Remember that GTP and ATP are equivalent in energy status. So this is essentially another ATP being burned. It just so happens that it's GTP. Once all those initiation factors are free and we have this transfer RNA bound, the 50S subunit can then sit down on top of the entire complex, and this would mark the end of the initiation process. Now, in eukaryotes, this is a very similar activity. These factors that are required, we call them EIF, for eukaryotic initiation factors. And the initiator transfer RNA is just simply a methionine transfer RNA. So it doesn't have to be specifically the N-formulated version. It's just a methionine transfer RNA. They're all the same. The start codon for eukaryotic translation is AUG. And not all AUGs can act as a start site. There are additional sequences that make the start recognition work. And this is because there is not a Shine-Dalgarno sequence. Instead, what we have is upstream, we have an AG that is approximately -3 to the +1 start codon, and then we have a G that is at the +4 position. But these are called Kozak's rules, named for the individual who first discovered them. But with this in mind, I want you to remember that in eukaryotic messenger RNAs, there is, in fact, a 5' cap, if you remember that. There's a specialized cap put on the end of messenger RNAs on the 5' end, and this AUG is typically the first AUG after the 5' cap. There can be some exceptions that there's always an exception, but typically that's what it is, and it will follow these particular rules. So initiation, you have a protein complex that we call eukaryotic initiation factor 4. This actually binds directly to that 5' cap. Okay. These are joined by a complex that is the 40S subunit, and this transfer the initiator transfer RNA, so methionine transfer RNA, and there are also some other initiation factors coming in as well. So this entire complex starts scanning along the surface of the messenger RNA, starting at the 5' end, and it stops when it hits the correct start codon. Once it finds that, it will pause there because it is binding to this AUG, because remember this complex includes this transfer RNA that has the anticodon. So it will join there, and then the 60S subunit will come in. And of course, most of these initiation factors are dislodged in this process. Now we can move on to the elongation stage. So this is the stage where we are adding amino acids onto a growing polypeptide chain, one at a time. The addition of each amino acid requires several steps, and they repeat in a cyclic fashion. Even though what you'll see is a very complex process, it can happen remarkably fast. So in bacteria, we can add 15 to 20 amino acids per second, and in eukaryotes, it's 1 to 6 amino acids per second. So even though bacteria are faster, they are both clipping along fairly quickly for what you'll see as a fairly complex process. So let's kind of walk through that. So here we have the entire complex, the entire ribosome with a messenger RNA in it. Right here in this example, we're showing that we are on codon 3, and we have a transfer RNA in the P site with its bound amino acid, and then we have the amino acids for codon 1 and 2 bound covalently to amino acid 3. The A site is empty. A new transfer RNA will come in that can base pair with the next codon. Now, this step of putting in a new transfer RNA into this A site is actually slightly more complicated than it appears. It actually requires what we call an elongation factor protein that actually hydrolyzes GTP in order to even fit it in here. So it is a slightly more complicated process, but regardless of that, we now have this in the site, and do note that we are burning more energy in this process. Then what happens is the growing polypeptide chain, which was attached to this transfer RNA covalently, is broken and transferred to this next amino acid. So this bond is replaced with a bond to this next amino acid. That transferase activity is actually carried out by a piece of RNA. So it is not a protein enzymatic process, it is an RNA enzymatic process. So the ribosome is in fact what we call a ribozyme, similar to what we see in splicing. All right, so it is then transferred over, and then another elongation factor will come in and facilitate the shift of the spent transfer RNA movement from the P site to the E site, and the movement of the growing chain and the transfer RNA from the A site to the P site. Again, that seems like a complex thing, and it does in fact require burning more energy. Once that takes place, we can then release the spent transfer RNA from the E site, and that sets us up to repeat that process over and over again. And this will continue until the point that we reach a stop codon. Now, translation is terminated once we reach that stop codon. And again, if you recall, there are 3 stop codons, UAG, UAA, and UGA. We also refer to these as nonsense codons because there is nothing that fits with it. There is no transfer RNA that goes in there. Now, since there is no transfer RNA, there are proteins that are called release factors that do fit into this space. And the 3-dimensional structure of these release factors is actually very similar to the 3-dimensional shape of transfer RNAs. So in bacteria, we have 3 release factors. There is an release factor 1, release factor 2, which recognize different codons. There is some overlap on UAA. And then there is an additional release factor, which isn't involved in recognizing the codon, but it is required for terminating the entire process and releasing all of the factors and releasing the ribosome itself. Now, in eukaryotes, there are only 2 release factors. There's just simply a single RF1, or ERF1, for eukaryotic release factor 1, and it recognizes all 3 stop codons. So 1 is equivalent to both 1 and 2 in bacteria. Eukaryotic release factor 3 is the equivalent of release factor 3 in bacteria. And again, it's required for that termination process. So here's a picture of that. We have the growing polypeptide chain attached to the transfer RNA in the P site. There is no transfer RNA to come into the A site. So instead, we get release factor 1 or 2, or RF1, in the case of eukaryotes, will fit into that space. That will facilitate the release. Well, number one, the cutting of the bond between the transfer RNA and the polypeptide chain, but also the release of both to fall out. And then the additional release factor will come in and disassemble the entire complex. And that is how we stop this process. And again, this release factor here has a very similar 3-dimensional shape to a transfer RNA. So let's take a second here to kind of compare and contrast bacterial and eukaryotic translation, and it mostly comes down to differences in factors, not necessarily differences in mechanics. So number one, in eukaryotes, we have this initiation factor 4, which is recognizing the 5' cap on messenger RNA. We don't have an equivalent in bacteria because we do not have a 5' cap. We have the initiation factors 1 and 3 that prevent the association of the large and small subunit until the messenger RNA is correctly inserted. We have these factors that are in eukaryotic systems. We have initiation factor 2 and its equivalent EIF2, which promotes the initiator transfer RNA being assembled onto the small ribosomal subunit with a messenger RNA. Eukaryotes also have this initiation factor 5, which is an additional thing that is needed for the dissociation of those initiation factors. In